session 2, I'm really proud to introduce is Professor Robyn Jamieson. So Robyn leads the iGenetics Research Unit at the Children's Medical Research Institute, and she also heads the iGenetics Clinic at the Children's Hospital Westmead. Alongside that, because she's not busy enough, she's also the head of the Western Sydney Genetics Program at the Children's Hospital Westmead and the head of Specialty of Genomic Medicine at the University of Sydney. And she'll be presenting today on what we've already had a couple of questions about, gene therapy for inherited retinal diseases. Thanks, Robyn.
So thanks, Eden, and thank you, Anna. It's a real pleasure to be here today. As you know, we've been in touch many times over the years to many of the people in this audience, and so it's really great to come together and learn even more about the inherited retinal diseases when there's so much happening in this space. So, as we've heard, precision medicine and the eye is a real place for this. There's these therapy opportunities that we've heard about. There's this amazing clinical and genetic heterogeneity that we've heard about that's been a real problem. But then there's genomic strategies now coming through that we can really understand the genetic causes and so therefore move towards the therapies that we've all been talking about. So when I started my lab 22 years ago, I try not to think about that number too much, but anyway, so it was just at the start of things, really. It was when the Human Genome Project was coming along and really showing us that amazingly we could be able to understand these genetic causes. And I had worked as a clinician and I'd also done a PhD in developmental biology, and I did functional genomics work in the UK. And I really thought that, there was a lot happening in the UK, that was true, but I really thought we had an amazing opportunity in Australia. We had amazing clinical services. So I was based at the Children's Hospital Westmead, which is now the Sydney Children's Hospitals Network, but also, of course, as we know, in Melbourne and all the other states, we also have wonderful clinical services. And then we also had the Children's Medical Research Institute, which was a great basic science research institute with incredible interest in understanding developmental biology, cell biology, and how these things worked. And then we had genomics coming along. And so I really thought this is an amazing opportunity to bring all that together. And I must admit, when I came back to CMRI in 2002, that was not necessarily seen as being a great way to move forward, because at that time, basic science was seen as being, fantastic, and that was correct. But the links to the patients and the families and how we could make that really sing and work was a little bit harder to see. And that was especially the case for eye diseases, genetic eye diseases. There were many, many different disease genes. How could we really get a handle on this? In 2004, I remember being, someone saying, how can we possibly do this? But really, genomics really came along and we could really find answers for patients. And so our work has always been very involved, the iGenetics Research Unit, in the clinical aspects of what we're seeing for patients, with the families and what the genomics is telling us, the family structure that Alan talked about, and then how we could work this in together with the functional genomics, which functional genomics is meaning, well, we've got these genes, but how do they work? How do they really cause the disease problems? And the treatment models that go with that. And as the technology has moved along, we have moved with the times. Lauren was talking about the AAV and the CRISPR-based strategies, which we've then also incorporated into our work. And then Anai has mentioned about the organoids, which then is really a valuable tool, as Anai has indicated, in terms of understanding the therapies. And of course, the mouse models can also be very useful as well. The whole point of this work has always been to be able to return this for the patients and the families in terms of the genetic variant identification, the diagnoses, the new treatments and the clinical trials. And it really is an amazing thing to see that over those 20 years we have been able to make such amazing progress in all of these areas. And there is so much more work to do, but I really think that we are really on the path. And so we were therefore very excited to be the first team to be able to deliver this clinical gene therapy in Australia. which was the one that I think John has mentioned and others have mentioned in their talks about RPE65 gene therapy. And this was one gene therapy for the RPE65 gene. And it was commenced in 2020. As you can see there, as with all of this work, as you've heard, it really takes a huge team. The basic science work takes a huge team. The clinical delivery work takes a huge team. There's many people here. There's a combination here to get this into the clinic. There's the pharmacy people, the operating theatre, the regulatory aspects, the genetics, the clinical ophthalmic diagnosis, and then the delivery of the actual therapy, which of course requires incredible skill as well. And so these are the first two patients in the image here. This is showing two patients. They're looking up, seeing the stars at night. So they had not previously been able to see the stars in the sky. And so this was, they were just incredibly excited and to go onto ABC television and tell everyone about that. So I just wanted to give you a little bit of an insight into the journey for that. It was something that was in clinical trial for a number of years. It was then approved in the US, approved in Europe, and then we did a lot of work with the company that was involved in this therapy, as well as all of the team that needed to be involved to get that across the line for public funding. So we really wanted this to be able to be approved so that the therapy could be used in Australia from a scientific and clinical point of view, but also that there could be public funding so that this would be available to patients that needed it, no matter what their economic background was. And we were able to achieve that, and so that meant that we could deliver the therapy. So these are the criteria for that particular therapy. Someone asked, I think, about RPE65 gene therapy and what was required. So it has to be the clinical diagnosis of an early onset retinal dystrophy or a Leber congenital amaurosis. There's particular clinical criteria. The genetic diagnosis has to be in place. Pathological biallelic variants, gene mutations in the RPE65 gene. So the pathway that Alan talked about is very important here in getting that genetic diagnosis. And then the ophthalmic work has to happen again in terms of are there sufficient viable retinal cells. So all of those processes that John talked about in terms of the clinical assessment is very important in terms of working out are there viable retinal cells for the gene therapy to actually go into to be able to make those those photoreceptor cells be able to work again. And so this gives an indication of, I think, some people in the room, if there's, and I've had a couple of questions already today about where people may go to be able to get on that path towards a genetic diagnosis. In Australia, as we know, we're very geographically dispersed, but we are fortunate to have local genetic services right across the state in New South Wales and also other states in Australia as well. To find your local genetic service, I've given the website here. You can go into that and that will give you a location for your genetic service throughout Australia. You can type in your suburb or your town, and then that will give you information about where your local genetic service is. And your GP can refer you to your local genetic service so that you can start the process to get that underway. And then, as Alan and John have mentioned, we really value the benefit of the ocular genomics multidisciplinary team meetings. So that brings together the various team members that we need to help in when we're getting down to the nitty-gritty, particularly if I'm talking about the example of the RPE65 gene therapy, getting down to the nitty-gritty of will we be able to offer this particular gene therapy for this patient? We have to go through things like checking on the clinical diagnosis, the genetic diagnosis, and then sometimes we have this problem of variants of uncertain significance, as Alan mentioned, in these conditions, which is really a difficult problem because what that means is that the TGA, so the regulatory authorities, the person has a variant in RPE65, but it's a variant of uncertain significance, and even though we might think clinically, this is really the case, we're going to be able to give this therapy, we can't. We're not allowed to from a regulatory perspective. So we are working, I'll talk about that in a moment, we are working on ways to get across that particular hurdle. And then other things we think about is we also use this team for all of the inherited retinal diseases and all of the genes, all of those over 250 genes. And so that gives a lot of process talking there. And the way that we've distributed this is by having the genetic services and the ophthalmologists in all those areas across the state and also nationally be able to refer to this. So that they can get that expert opinion about the particular condition and then take it back to the patient. There's no way, with the number of patients that we have, as Alan's indicated, approximately 1 in 1,000 people affected with these conditions, that our services are not set up that every person can necessarily be seen individually at a multidisciplinary clinic, but we can do this by doing this virtual multidisciplinary meeting approach, and that's really improving equity of access for people across Australia to get more of an understanding of the conditions. And then, as Anai has talked about, the whole retinal organoid space. So we realised that being able to investigate these genes and processes in a human model would be a fabulous thing because, of course, up until that stage, people were working on animal models, mostly other cell-based models. And so clearly being able to work in the stem cell space where we can get these to retinal organoids, which are then patient-derived materials and being able that really then mimic really well the photoreceptors and so on, as Anai has mentioned, is really a fabulous thing. And so we were really pleased when Anai was able to join us in Sydney around about 2019, I think it was, and really progress this field. And so members of my team who are here today, so Tu-Ha Loy and Reva, sorry Reva, I put your name there incorrectly, but Reva is here working on this space as well and able to generate these retinal organoids as we've mentioned. And one of the really valuable aspects of this is to help us with the variants of uncertain significance. I mentioned that this is a real bottleneck to genetic diagnosis in some cases. And so this was a particular family that we've seen in Sydney who had 2 variants in the RPE65 gene, one from each parent. And one of them was a clear-cut pathological variant, but the other one was a variant of uncertain significance. And so what this meant was that even though we thought that they had the clinical features and they had enough viable cells, that they would not be suitable for the RPE65 gene therapy. In this case, that particular variant of uncertain significance was at a region of the gene where we thought it might be doing what's called a splicing abnormality. And to investigate splicing abnormalities more, you actually have to look in the tissue where that gene is actually expressed. And RPE65 is not expressed in the blood. So normally when we're trying to look at splicing problems, we try and take a person's blood or just a skin sample, look in their skin cells to see if there's a problem with the splicing. But there wasn't, RPE65 isn't expressed in those cells. So therefore we had to go down the stem cell route and take a sample from the patient, turn that into the induced pluripotent stem cells, and Anai has explained the process for that. And then we were able to look at that particular gene and its expression pattern in what's called the retinal pigment epithelial cells, which are the supporting cells, which is where the RPE65 gene is expressed and causes all its problems when it doesn't work properly. And we found a clear-cut abnormality in that kind of way. We're a research lab, the iGenetics Research Unit is a research lab, so we then have to take that to the clinical diagnostic lab, with whom we work closely at the Children's Hospital at Westmead, and present our evidence to them, and they are then a NATA accredited diagnostic lab. They can agree with us or they can disagree with us, but in this particular circumstance, we had very clear-cut evidence, and so then they agreed that that was a pathological variant. And then those 2 children were eligible for the RPE65 gene therapy and have, in fact, been able to have those. and had that therapy, and that's really improved their vision, as I'll talk about. And so Ben Nash, who was a PhD student in my lab and is now working at the Children's Hospital Westmead in the diagnostic lab, was really integral to that work. And Tuhar Roy, who's a senior postdoc in my laboratory, who's also integral to this work, that made a real difference for those patients. So just to tell you, once we get the diagnostic sorted and we get the clinical retinal information, and we think all good, we can go with this patient to take them to gene therapy. To get that on the road, there was really an enormous amount of work that had to happen from a regulatory perspective. Of course, we had to have the patient identification, the genetics, the orthoptics, and the ophthalmic assessments. We had that pretty much sorted by that stage, but we have to get the therapeutics TGA approval, Therapeutics Goods Administration approval for any particular gene therapy. And this is what we had to do with Luxturna, and so we now, which is the RP65 gene therapy, we're now looking down this path for other gene therapies that are coming through. So John mentioned RPGR as a particular gene that we're really hoping is going to go this path, that the Luxturna gene therapy has been able to go. And so we have to think about all these processes. So how do we make sure we can get the TGA approval? There's the Office of Gene Technology Regulator, because this is a viral vector therapy. They want to know that we've got all the structures in place for that. There's the pharmacy requirements, the nursing and surgical requirements, the operating microscope, the workforce development, the ophthalmic equipment, and all of the funding arrangements. So of course we want to get public funding for these conditions as well. So just to show you, so there's a lot of work in the hospital then in terms of the quality assurance. And I talked about trying to get the stem cell therapy to this kind of space. So we have travelled this path with gene therapy and all of the quality assurance and so on that's expected for a clinical therapy. And all of the particular laboratory requirements that are there. And then on surgery day itself, the preparation of that drug for delivery on that day. There's a lot of regulation and making sure that the pharmacy is doing everything correctly. They need two operators to prepare the drug for that day. The drug is normally held at minus 80 degrees Celsius, so it's really deep frozen. So and that's the way it's stable. So you can't defrost it. Once you've defrosted it, it has to be used. So there's kind of a lot of pressure on this because it is an expensive therapy. And so it's actually 2 doses that are drawn up in case one is dropped or something like that. And they do a lot of work to get that exactly right. And then that's delivered directly to the operating theatre, and there's a 4-hour expiry. It has to be delivered in that time frame. And on the particular day, the patient has an anaesthetic. It's a subretinal gene therapy delivery. The 2 syringes, as I mentioned, the primary and the backup. That 4-hour administration window, pressure's on, so it just goes ahead with that way. There's very fancy operating equipment delivering to the back of the retina, as Lauren mentioned, and the bleb is formed. You can see there at the back of the retina, the bleb is formed, that little retinal detachment that then subsequently gets resolved. And just quickly about the outcome measures. So the patients, generally speaking, have certainly been improved from this therapy. This is the Luxturna RP65 gene therapy. They've had improvements in their retinal sensitivity, which is called an FST test. And the government wants a pre-therapy test of that, post-therapy test of that, to pay the company. So there's quite a lot of regulation around this. The patients, generally speaking, have improved visual fields, so they can see better peripherally, and some have certainly had improvements in their visual acuity, and all of them have mentioned that they can now see stars at night, which they couldn't before, and all of them have mentioned mobility benefits. For instance, they don't sit in the wrong seat in the train, or they don't sit on someone inadvertently, or they can see their partner's face more clearly for now than they ever could before. So it has certainly been beneficial for the patients. Just the other thing that I want to give a shout out about as well is that an important thing that came up in terms of getting this across the line in terms of getting the government to approve this and also to have the argument for the public funding for this therapy was the health economic impacts. Now, this is really annoying, I know, because from a patient perspective and a when we see patients, what we really want to do is be able to improve the vision and the situation for them. But unfortunately, when government gets involved, health economics comes into it. I guess that's a reality which we have to live. And so we embarked then on a study because we realised this was going to be an issue, looking at the healthcare and societal costs and the impact of inherited retinal diseases on people, not only from whether they went to the doctor more often, but what were the impacts for them in terms of their life, in terms of did they lose their job when they shouldn't have, or were there real those economic impacts, but also is there a higher risk of people having car accidents or walking onto the road and that sort of thing. What is the healthcare and societal costs of that, so that we can have an argument to balance that against the benefits of the therapy. And so that actually requires quite a bit of modelling. We actually did work with an agency initially who said, that's just in children, that kind of doesn't matter somehow. They didn't sort of see the lifetime impact of those conditions, which is certainly something that we see when we talk to people affected and the families. And so we have worked with Deborah Schofield at Macquarie University in a collaboration with her to have a modelling process where we can look at that over the life years and then look at the costs and the overall lifetime costs. So we have had this paper published in the Medical Journal of Australia in the middle of last year, is estimated to be, on the basis of the study that we did, which was about over 100 patients in that study, around about 5.2 million lifetime cost for patients and carers. And then interestingly, approximately 87% of that societal costs and only 13% healthcare costs. And this is another problem that IRDs run into in terms of some conditions cause you to go to hospital and have a lot of hospital costs. And so those are easier for government to kind of assess and see. Whereas these societal costs can be a little bit hidden because they're the burden that each individual and their families are taking on. And so we really wanted to draw that out. And I know some of you have kindly been involved in the surveys that go with this study, which are in depth and take a lot of time. I know that, but this is the reason that we're doing it, because we really want to get across to government. This is real impact for people, and so we really need to be advocating for the funding that we need for these therapies. And then the total cost of IRDs per year, approximately 1.5 billion. So this was a study looking at inherited retinal diseases more generally. As other gene therapies are coming online related to particular genes, we'll be looking at more specific focused areas because we think that government will want that information as specific therapies are coming through and we're trying to get public funding for them. So as with the study that Eden has mentioned, with this study, if you are approached, please consider being involved in these studies because they're really, really important. Government really wants to hear the patient voice, the family voice, to get these things so that we can have therapies for people as we want them. Because as I said, this is and as Alan has indicated, this is where we want to be. This was the whole aim of the research, that we have the teams doing the work, the multidisciplinary team working with that, and then our pathways in terms of whether it's an internationally approved therapy, a clinical trial, or an investigator-led approach to get to the clinical therapies and clinical trials for people affected with these conditions. And I really want to acknowledge the people involved. As I said, there's a lot of laboratory work. This is my laboratory team here, and they slog it out. There's a lot of work with laboratory work. Hours and hours in the lab, looking after the cells, as Anai mentioned. This is not easy work, and they are very dedicated. to really trying to help in this field. And then, of course, the hospital team as well. So the vitreoretinal surgeons, the pharmacy people, the nursing people, it's a real team effort to really get that going. And so I really want to pay tribute to all of the people involved and who are really helping towards this work. And I really hope that we can work towards more advocacy for these conditions. And Leighton and Rosemary Boyd from Retinal Australia and Julia are here, and great to see you, and all of the other people who advocate for these conditions. The Usher Kids team are here as well. And so, this is really good because government really listens to people with these conditions, and they really want to hear your voice as well. So thank you very much, and look forward to chatting further.